Hello. Welcome to SRX Creates. So this is our third part of level organizations of life. In this chapter, we are going to deal with the chapter cell, which is the basic unit of our life, from where its tissues are going to form tissue system, organ system, and organism finally. Now, in our last lecture, while starting the lecture, the starting paragraph includes a word which was organelle. We are going to deal with this organelle in this chapter. Okay, now let's see. There are innumerable species, a lots of species we have. Now some of them are unicellular, multicellular, but a living body is made up of a cell. Let it be uni or multi, and it is the structural and functional unit of life which is also replicating its mass or gets divided. It's replicating or divided. Now replicating means what? To form a replica. This is a cell. After its division, it gives us two cells. 2 gives more 2. Like this, many numerous cells forms, and the basic unit cell makes us the human body. Then tissue, cell to tissue system, etc. Okay. Now this was first discovered by Robert Hooke. Okay, Robert Hooke discovered a cork cell which was dead, all of course. Now with the discovery of electron microscope, Knoll and Ruska given new dimension to cells. Now the cell has prokaryotic and eukaryotic. There are two types of cell. Now in eukaryotic, which is the more developed one, we have cell wall, cell membrane, cytoplasm, nucleus, mitochondria, plastid, endoplasmic reticulum, Golgi body, lysosome, vacuole, ribosome, centriole, microtubule. These are the cell organelles. These are our cell organelles. Now we will deal with the particular organelles one by one. Now see one thing. This diagram is of the animal cell and this is the plant cell. This animal cell and plant cell are very important for 5 marks diagram with function question. Okay, you all have a 5 marks question at last in the 40 marks first summative question paper, where you will get this question as draw the diagram and the labeling will be given. You can't choose the labeling. Any 4 labelings will be there. 2 for labeling, 3 for the drawing. Means half into 4, 2, and 3 for the drawing. Neatness especially. Now cell wall. This cell wall is a wall which is a thick and rigid permeable coat. Means permeable means through where which something can pass through. Now outer means which is outside, and plant cell formed of cellulose is called cell wall. Means the ingredient is cellulose here. Okay. Now structure. This is formed of middle lamella, primary wall, secondary wall. Now in this picture, carefully see that the primary wall is the outside. This is the outside. Secondary is next to primary, and next to secondary there is middle lamella, and next to middle lamella there is again a layer, after which we get primary wall, plasma membrane. Now in this cell wall structure we see pore like structure which is called plasmodesmata. Okay, and a nucleus and a vacuole, and this fluid semi-fluid matrix or structure is called cytoplasm. Okay, I think basic structure is clear. And where is middle lamella, where is secondary wall, where is primary wall, it's clear. The middle lamella is formed of calcium, magnesium, pectate, and several proteins. Okay, this is the ingredient, and it is between 2 plant cells. Means if there is one more plant cell, middle lamella, let's say this is one more plant cell. Okay, so it helps to join 2 or more cells together, like between the 2 plant cells. The accumulated intercellular matrix represents middle lamella. Okay. Now primary wall is at the two sides of middle lamella. See, there are two primary walls, this portion and this portion, two primary walls and one middle lamella. Clear? And it is the outermost investment, means here is layering, and see, it is the outermost. Clear? And it is thin and permeable. Thin, you know, it's thin, and permeable means it can pass through. Now composed of cellulose, composition is cellulose, may contain hemicellulose, lignin, suberin, and yeast has primary wall made up of chitin. The secondary wall is thick, rigid, permeable, and contains three layers of cellulose, hemicellulose, lignin. Okay, and it is present inner to primary walls, means next to primary wall. Okay, I think these are clear. Now the functions of cell wall are, it protects protoplasm. As you can see, if this is a cell wall portion, it is protecting this intercellular matrix. So see, this is the cell wall and it is protecting this intercellular matrix, so it works as the protection of protoplasm and plasma membrane, right? Now it provides mechanical strength. If the cell wall doesn't have its wall, if our house doesn't have its wall, then is it not like very tough? Does it get its strength while there is a storm, there is a rain, right? So it gives mechanical strength. Contributes shape. Without a wall, does a building get its shape? No, right? So for shaping it. Now movement of water, if that is why the word permeable is there. See, water and ions, if it can't pass through, if it's not permeable, how can water and ions pass through? And plasmodesmata, this is plasmodesmata which helps in intercellular connections. Connection means if pores, so material from one cell to another cell can pass through. As I said in the last lecture, that inter means international, international means there are two countries means two different cells and they will have their connection. Intra means there will be one cell with which connection will be built up. Now plasma membrane. Now this is the membrane portion. We are talking about this portion, this plasma membrane, which is bounding the cytoplasm. See, every living cell enclosed its protoplasmic mass within an elastic. Elastic means which can be stretchable. Semi-permeable means it can permit few things but not all the things, like permeable things. Okay, it's called cell permeable. Dynamic means it can change its shape. Okay, needs ultrastructure. It forms of two layer, the three layer, two outer and one inner. Our inner layer is formed of lipid, outer layer is formed of protein. Each lipid leaflet contain two polar ends. See, two polar ends. One composed of phospholipid and other is formed of fatty acid chain, non-polar tail, non-polar tail, okay, non-polar tail. This is the lipid one, this is the protein one. Okay, see, the chemical composition is plasma membrane is 40% lipid, 50% protein, 10% carbohydrate. Now there is one word, the lipid polar head is hydrophobic and non-polar tail is hydrophilic, and philic means loving, so they love water, means this polar head loves water, and these non-polar end is hydrophobic, means it hates water, so it's inside and these are outer, so these loves water. Okay, this is a lipid bilayer, means two layers, lipid bilayer. Okay, done. In the protein layers there are several enzymes, sodium, potassium. Okay, clear. Now what's the function? See, it helps in protecting the cellular living portion, means the cytoplasm. Acts as an osmotic barrier, means osmosis nahi hoga udhar, and contributes in the formation of cell organelle and nuclear membrane, means formation of cellular organelle will be there only inside the cell cytoplasm, inside that plasma membrane portion. It facilitates diffusion and osmosis, means to high concentration to low concentration, something get passed through the layer. Promotes intake of liquid and solid food, so external medium. Phagocytosis. Now what is phagocytosis? This is a cell, there is a food, this cell now, after this getting the food, it becomes like this. It grabs the food, then after grabbing the food it encloses it, then finally it be like this, takes the nutrition. This is phagocytosis. Okay, phagocytosis done. Now cytoplasm. What is cytoplasm? Cytoplasm is the fluid that fills the cell. Scientists used to call the fluid protoplasm. Okay, this protoplasm is conventionally divided into cytoplasm and nucleoplasm. Fluid is called cytosol, and nucleoplasm is the fluid in the nucleus. Okay, the cell organelles are suspended in the cytosol. Okay, the cell organelles, as I said in here, that they are suspended in the cytosol. Cytoplasm has many different molecules dissolved in its solution. You will find enzymes, fatty acid, sugar, amino acid. We all learned these terms in our last lecture. Waste products are also dissolved there before they are taken by vacuoles or sent out of the cell. There is also excretion system. Cytoplasm is a thick solution, that fills each cells, enclosed by cell membrane, mainly composed of water, salts, protein. In eukaryotic cell, the cytoplasm includes all of the material inside the cell and outside the nucleus. Okay. Most of the important activities of the cell occur in cytoplasm. Function 1. Cytoplasm contains molecules such as enzyme responsible for breaking down waste. 2. And help in metabolic activity. 3. So there are 3 functions. Nucleus. Nucleus is the control center. It can be also the brain. Okay. It is the largest organelle. Okay. It contains DNA, means the genetic material. DNA is deoxyribonucleic acid. What is it? Contains all the information of the cells to live, perform their actions, and reproduce. Not all cells have nucleus. Biology breaks cell types into eukaryotic with a defined nucleus and prokaryotic with a false nucleus or a non-defined nucleus. If you don't have a defined nucleus, your DNA is probably floating around. Means the DNA is not enclosed anywhere. It is available in this kind of way. This is a folded structure which is called nucleoid, which also has a genetic material which is floating, not stuck in a position. Now, defined nucleus that holds the genetic code is advanced feature. Okay, these things that make a eukaryotic cells, like having defined nucleus and other organelles. Okay, the nuclear envelope surrounds the nucleus and all of its contents. The nuclear envelope surrounds this all contents that live in a cell. Now this nuclear envelope is a membrane. Nuclear envelope is a membrane like structure covering wall like structure similar to cell membrane. Cell membrane and nuclear membrane are similar, just cell membrane covers the cell and nuclear membrane covers the nucleus around the whole cell. When the cell is in resting state, is something called chromatin. Okay, chromatin is a term when the cell is in resting state. And chromatin is made up of what? DNA, RNA, and nuclear protein. Composition of chromatin is DNA, RNA, and nuclear protein. DNA, RNA, and nucleic acids. We all know chromatin becomes very compact when cell is going to divide. When does chromatin becomes very compact? When cell is going to divide, it condenses. Condense means to stay more stuck, more compact. When the chromatin fibers comes together, you can see the chromosomes. Means when they come more together, like this structure forms. Okay. Nucleolus is also inside of the nucleus. It is made up of RNA of protein. Okay. Now mitochondria is the powerhouse of the cell. Everyone knows that. And mitochondria organelles acts like a digestive system of the cells. Digestive system means, see, cell has their own major organs for performing all the major functions of a cell, like our organ system. Okay, so it takes the nutrients, break them down, create energy, then possess, creating cell energy. As cellular respiration also helps in respiration also. Now, most of the chemical reactions in the cellular respiration happen in mitochondria. Means this is the main center of reactions. Mitochondria is shaped perfectly to maximize its efforts. Means it can do more and more work than it can. As in other organelles, the outer membrane covers the organelle. See, mitochondria has these different organelles also. See, the outer membrane covers the organelles and contains it. The inner membrane folds many times. See, this is folded many times. These foldings are called cristae. The folding increases the surface area. See, surface area जितना बढ़ेगा इतना मुझे अंदर का स्पेस मिलेगा, उतना कॉम्पैक्ट भी रह पाऊंगा और स्पेस मुझे जब लगेगा मैं उसको खींच सकता हूँ. So it can fold over many times. Folding increases, so surface area increases. Many of the chemical reaction happen in the inner membrane of the mitochondria, so it can be also said that cristae takes part in all major of the reactions happening in mitochondria. The increased surface area all allows the small organelle to do as much of work. Increased surface area, see, it helps to do more work, and the fluid in these foldings is called matrix. Plastids are the plastids, okay, plastid. Plastids are the site of manufacture and storage of important chemical compounds used by the cells. Means it is the manufacturer. Plastid often contains pigments. Pigment means colored substances in which is helped in photosynthesis. Pigments which can determine cells color. Now they are broadly classified. Then this pigment is classified into chromoplast and leucoplast. And chromoplast is divided into chloroplast, rhodoplast, pheoplast. Now pheoplast is for dark brown color, chloroplast we all know green color, and rhodo means red color. Leucoplasts are non-living, not leucoplasts are non-pigmented plastids, means there are no pigmentation, no coloring. Leuco means white and plast means living. Their main function is to store food material, means plastids store food material. Okay. Chloroplasts are the food producer, means they produce food. They are found in only plant cell, not animal cell. Animals do not have chloroplast. Every green plant you see is working to convert the energy of the sun into sugars. They create sugar as a byproduct by the process in the oxygen where we breathe. The process happens in the chloroplast. Okay. Two membranes protect the inner parts of, see, there are two membranes. Okay. The stroma is an area inside of the chloroplast where reactions occur and starch and sugars are created. Starch, sugar are created. Okay. One thylakoid tract is called granum. See, this one thylakoid tract, these thylakoid. These three thylakoid together is called granum. This is called the inner membrane. This free portion is called stroma. This is between two membranes, outer and inner membrane. This is called the intermembrane space. Two different membrane, so there it is different here, so this is called intermembrane. This is thylakoid, and by which two granums are connected is called lamella, and in the thylakoid there is a space called lumen. Okay, done. I explained this portion, I am not reading this stuff as I explained it here. Now endoplasmic reticulum. Endoplasmic reticulum is another important organelle. Okay, it is also often said as ER. ER is a packaging system. Okay, packager. Does not work alone. It takes with Golgi apparatus, ribosome, RNA, Golgi apparatus or Golgi body. It creates a network of membranes. Network means creates a network. So does the packaging system. Like there is a courier facility, hub. In Amazon we see now. Then there are two types of ER, rough ER and smooth ER. They have the same types of membrane. They have different shapes, and a rough ER. They have different shapes, and rough ER has ribosome attached. Okay. They have different shapes, and rough ER means different shape means here what? Different shapes means they can be circle, square, rectangle, any. And rough ER. Rough ER means rough and smooth ER. Rough means here is packaging of proteins, and smooth means storage of steroid. Steroid is a type of substance which we found in human body, of course. Now smooth ER is has its purpose in the cell. Okay, and it acts as storage organelle, means smooth acts as storage and rough acts as packaging. When a protein is complete, rough ER pinches off a vesicle. Vesicle means like inflammation is seen, like this is the organelle and inflammation is in this is a vesicle portion. Okay, now this vesicle is a small membrane bubble like structure. Okay, and can move to Golgi apparatus. Okay, or cell membrane. And rough ER looks like sheets of bumpy membranes. Means bumpy means like this. And while smooth like tubes. See, tubes. And rough is C, bumpy. Okay. Now Golgi apparatus, which is found in most of the cells, is a packaging organelle with ER, has a profound role in secretion and was named after Camillo Golgi. Now Golgi complex gathers simple molecules, simple, and combines them to make more complex. These takes big molecules, packages them into vesicles. Big molecules, then is C. This is the vesicle, as I said, vesicle, and either stores them or sends them out of the cell. Means if they require it, store it. Then if not required, they just leave it out. Then the Golgi complex works closely with ER, rough ER, works closely with rough ER. When a protein is made in the ER, something called a transition vesicle is made, means which is just forming the transition position between formed and unformed. The vesicle or sac floats in the cytoplasm to the Golgi apparatus and is absorbed. Means the cytoplasm absorbs this vesicle like thing. After the Golgi does its work on the molecule or the sac, a secretory vesicle is formed and released into the cytoplasm, and the vesicle moves to the cell membranes and the molecules are released out. Means to release out this molecule, this vesicle type organelle is formed here. Okay, bubble like structure. You will find organelles like lysosomes. Lysosomes are often called as suicidal bags. Suicidal bags. Okay, nearly every animal like eukaryotic cell has this. A lysosome holds enzyme that were created by the cell. Okay, purpose is to digest everything. They might use to digest food or break down when the cell break down the cell when it dies. See, that is why it's called the suicidal bag. Since lysosomes are little digestion machines, they go to work and cell and absorbs and eat some food. Once the material is inside the cell, the lysosome attach and release their enzymes. The enzymes break down complex molecule and includes complex sugars. Means it helps in the nutrient absorption reduction in the cell. Okay. Vacuoles. Okay. Vacuoles are storage bubbles. They found in both animal and plant, but in animal these are small, but in plant these are very big. They might store food or any variety of nutrients that a cell might need to survive. They even store waste products also, and to help the cell protect from contamination or waste products. Those waste products cell sent out of the cell also. See, this is vacuole, very big in plant cell. The structure of the vacuoles. Wait. Structure of the vacuoles are fairly very simple. There is a membrane surrounds the mass of a fluid. In that fluid there are nutrients or waste products, may also contain water also. In plant cell the vacuoles are much larger than in animal cell. See, I have said you, when a plant cell has stopped growing, there is a usually one very large vacuole. Those vacuoles gain or lose water depending on how much water is available to the plant. A drooping plant has much lost of its water, so vacuoles are shrinking. Means when the water is less in the plant body, so it will shrink, and the water is there in a good quantity, so it will not shrink. Maintains a basic structure because of the cell walls. When the plant finds a new source of water, the vacuoles are refilled again. Okay. Now ribosomes. Ribosomes are need to make proteins. They're protein house. Now they might use as enzymes also. The ribosome is just not a piece. See, there are two pieces. Two pieces or two subunits. Means if I say 50s or 30s, these are two subunits of ribosome. Okay, there are 60s and 40s. Smaller is 40 or 30, and this is 60 or 50. When the cell needs to make protein, mRNA, M is type of RNA, mRNA is created in the nucleus, and mRNA is sent into a cell. C, created in nucleus, sent to the cell, and the ribosomes also. When it is time to make the protein and the two subunits come together and combine with the mRNA, when it's the correct time, the subunits lock onto the mRNA and starts protein synthesis. When they start protein synthesis, they lock the subunits. Lock. See, lock. 60s and 40s models works fine for eukaryotic cells. Prokaryotic cells have ribosomes which has 50s and 30s. 50s and 30s is a type subunits which has only prokaryotes, contained in prokaryotes only. Now this S is also known as Swiftberg unit. Okay, sedimentation rate. Done. Now centriole. Centriole. Many microtubules to centriole. Centriole has two small organelles. Every animal like cell has two small organelles, right? Those are called centrioles. They are there to help the cell when it comes to divide. Okay, it helps in making spindle fiber. You will usually find them near the nucleus, but they cannot be seen when the cell is not dividing. When the cell means जब cell division नहीं करने जाएगा तब तुमको उससे दिखाई नहीं देगा. And the central is a set of microtubules. See, microtubule. There are 9 groups of microtubule. When two centrioles are found next to each other, they are at only 90 degrees, means right angle. And centrioles are found in pair and move towards the pole when the nucleus, when times spindle fiber means, see, you do not know what is cell division, but I am just saying if this is a cell, there is a central portion. They formed a spindle-like structure, spindle fiber, and chromosome are there. Chromosome. Okay, after this some chromosome go here, some chromosome get here, two cells are formed. Okay, just in simple. Microtubules are what? Proteinaceous and very filamentous, means many broke into many filament like structure, filament. And in plant and animal cell both are present, controlling the mobility, means it can help to mobile location, it can change its location, are called microtubules. See, spindle fiber, this is what I was saying, spindle microtubule or spindle fiber. Now the function is helps in locomotion, beating of cilia and flagella. During cell division, they promote the formation of spindle apparatus, as I said, and helps in intercellular transport of water minerals and takes a significant role. Okay, this is not required. Now, prokaryotic cells and eukaryotic cells, which is the types of cell. You can also draw this type of cells also. These are very easy, I think. Prokaryotic cells. Pro means primitive and karyon means primitive nucleus, means false nucleus or no organized nucleus, and found in what? Bacterial cell, blue green algae, anabaena, nostoc, glycoplasma. The cells which contain organized nucleus containing chromosome with basic proteins and membrane bound organelles are called eukaryotic cells. Okay, now very important, learn. Cell organelle devoid. Cell is devoid of membrane bound cell organelle. Membrane bound cell organelle is not found in prokaryotic cell but present in eukaryotic. 70s type in prokaryotic, 80s type in eukaryotic. 80 doesn't mean 40 plus 40. Okay, present and formed of murine. Cell wall is formed of murine, mucopeptide and peptidoglycan in prokaryotic, and formed of cellulose in eukaryotic. Chromosome is not formed in prokaryotic but formed during cell division in eukaryotic. Cell division is amitotic in prokaryotic, and but mitosis and meiosis is two types of cell division processes which is seen in eukaryotic cell. Nuclear memorial is primitive or not organized in prokaryotic cell, but organized typical nucleus and with nuclear membrane is seen in eukaryotic cell. Now, plant cell has plastid, vacuoles, cell wall, centrosome, lysosome. An animal cell has cell membrane, centrosome, lysosome, but without cell wall and plastid. See, these are the premium characteristic which is only present in plant cell and animal cell. See, plant cell, animal cell. This is a eukaryotic cell and prokaryotic cell. Okay, I was very sorry. I am sucking with my grammar. Sorry. I am very sorry that I said you to draw this structure. This is eukaryotic and prokaryotic cell. Draw this when you are asked to do it. Okay, very important. This can also come in the examination rather than plants and animal cell. Plants and animal cell is this. Now, differentiation between plant cell and animal cell. See, these are just present absent. Okay, so we complete cell here. Thank you for watching. Bye bye.